Every algorithm, compression standard, and digital display you look at is built around a single engineering bottleneck: the physical limitations of the human eye. This cross-section charts the anatomy of our biological receiver. Light-sensitive receptors aren't distributed uniformly. The vast majority densely pack directly at the fovea. There are two distinct types. Longer, slender rods process scotopic, low-light vision. Shorter, thicker cones handle photopic vision. This graph maps their log relative sensitivity. The distinct upper curve belongs to the rods, demonstrating that they operate with a vastly higher sensitivity to light than the cones plotted on the lower curve. Looking at this plot of relative response across the 400 to 700 nanometer optical spectrum, we see the absorption characteristics. There is significant overlap between the red and green regions, and a noticeably lower relative sensitivity to blue light. Because the biological hardware absorbs light unevenly across both physical space and the electromagnetic spectrum, processing visual data uniformly is a waste of computing power. An efficient digital system maps its data strictly to what these exact sensors can actually detect. It's logical to assume that doubling a light source's physical brightness makes it appear twice as bright to the human eye. But the eye's response to changes in illumination intensity is strictly nonlinear. This chart plots contrast sensitivity by measuring intensity change, delta I, against a background, I. The top right plot shows a constant 2% ratio, the Weber fraction. But the bottom right graph reveals varying curves when background levels are included, proving our response is entirely nonlinear. To accurately model the biological response, we must calculate in logarithms. Taking the derivative of the logarithm of intensity yields the exact ratio observed in the data, delta I over I. The math confirms the eye operates logarithmically. Because human contrast perception is logarithmic, digital processors encode intensity nonlinearly. Storing linear changes in brightness would mean allocating memory to data variations that the human eye physically cannot see. The eye's nonlinear behavior extends into spatial relationships. Identical gray squares will actually appear to be different shades depending on the brightness of their surrounding backgrounds, a phenomenon called simultaneous contrast. The physiological mechanism causing this is lateral inhibition. A neural signal traveling from the retina is not a one-to-one pixel map. It is a weighted combination of spatially adjacent receptors. Mechanically, illumination of receptor A alone creates a large signal. But if we add illumination to a neighboring receptor B, a signal transmits laterally across the retina to suppress the firing of receptor A. In strict engineering terms, these inhibitory weighting values represent the impulse response of the human visual system beyond the retina. The eye acts as an active spatial filter. This active filtering creates direct visual consequences, like the Mach-band effect. The edge of each uniform gray bar appears slightly darker on the left and lighter on the right. The visual system locally enhances edges where none exist in the raw data. To quantify exactly which spatial frequencies trigger these edge enhancements, researchers plot the modulation transfer function, or MTF, mapping the limits of human spatial perception. The MTF proves that the biological sensor prioritizes sharp boundaries over smooth gradients. Digital algorithms take advantage of this by manipulating images in the frequency domain, directly addressing the spatial patterns the eye filters for. To apply this frequency domain mathematics, the continuous analog distribution of real-world light has to be captured and reduced to a sequence of discrete numbers. We achieve this through 2D spatial sampling. The system reads the continuous luminance values across a regular grid, taking discrete measurements at precise spatial steps, denoted as x and y. This creates a strict mathematical consequence. When you sample a continuous space signal, the resulting discrete space spectrum becomes a periodic replication of the original signal spectrum, tiling infinitely in the frequency domain. To maintain the integrity of that tiling without the frequencies overlapping and corrupting the image, a phenomenon known as aliasing, the sampling rate is rigorously bounded by the Nyquist-Shannon theorem. Once sampled, the continuous amplitude of the light is mapped into a finite number of discrete levels. This is quantization. A 24-bit true color system, for example, quantizes the spectrum into roughly 16.8 million exact color levels. Forcing a continuous reality into digital buckets requires either truncation or rounding. That necessary approximation introduces quantization noise into the final data array. Because the act of sampling and quantizing alters the signal, producing periodic spectral artifacts and noise, we require robust linear algebra to control the image in its discrete form. That control relies on the two-dimensional discrete Fourier transform. This mathematical engine isolates the specific spatial frequencies that our biological MTF pays attention to. The 2D DFT calculates the frequency domain matrix by multiplying spatial pixels against a forward transformation kernel. Because this kernel is separable, separating the variables lets processors run calculations on rows first, then columns, drastically reducing computational load. The 2D DFT is defined mathematically by a unitary matrix. By definition, a matrix is unitary if its inverse is exactly equal to its conjugate transpose. This means the columns of an n by n unitary matrix are orthonormal. Together, they form a complete, mathematically sound set of basis vectors to describe every point in the n-dimensional space. Therefore, the discrete Fourier transform is a completely reversible linear transformation. It mathematically remaps the image from spatial pixels into a matrix of frequencies without losing or destroying any of the underlying data. Geometrically, a unitary matrix simply performs a rigid rotation of the coordinate system around a stationary vector. Parseval's theorem confirms that during this rotation, the total energy of that vector is perfectly preserved. Spatial energy equals frequency energy. This enables the most valuable engineering feature of the unitary transform, energy compaction. When the spatial image is remapped, the transform packs a massive fraction of the visual energy into relatively few coefficients clustered close to the origin. The relevant image data is concentrated entirely into the low spatial frequencies. Now, return to the biological constraints of the human eye. The retina's lateral inhibition and the modulation transfer function make us highly sensitive to specific low-frequency contrast patterns, but effectively blind to high-frequency visual noise. By mathematically compacting the energy into the low frequencies, the 2D DFT isolates the exact data the biological sensor requires. We can heavily compress or entirely discard the remaining high-frequency coefficients because the human eye is physically incapable of perceiving them anyway. The pure mathematics of the unitary transform perfectly mirror the physiological limits of human biology.